Well, thanks, everyone. This is a new initiative from the AANS and the CNS tumor section on behalf of the Early Career Neurosurgery Committee entitled Latest Highlights in Neurosurgical Oncology. And we're going to be starting with webinars twice a month. Everyone is invited. And our first speaker is going to be Quinn Ostrom, who received her PhD in epidemiology and biostatistics in 2018 from Case Western Reserve University in Cleveland, Ohio. She completed postdoctoral training in integrative cancer epidemiology at Baylor College of Medicine in Houston, Texas. And she's currently an associate professor in the Division of Neuroepidemiology within the Department of Neurosurgery at Duke University School of Medicine in Durham, North Carolina. Dr. Ostrom is scientific co-primary investigator of the Central Brain Tumor Registry of the United States, the largest aggregation of data on newly diagnosed brain and CNS tumors in the world, and the primary source for brain and CNS cancer statistics in the United States. Dr. Ostrom research program focuses on identifying risk factors for primary and metastatic brain tumors in children and adults using novel data sources and analytic techniques. Her talk today is going to be entitled Searching for a Cause for Glioma, Population, Risk Factors, and Genetics. And without further ado, please welcome Dr. Quinn Ostrom. Thank you.
Thank you so much for the invitation. I'm excited to talk to you a bit today about glioma epidemiology. So just to put brain tumors into the broader context of cancer, which I imagine I really don't need to do for this group. So they are relatively rare in comparison to the most common cancers in the United States. But when you account for both benign brain tumors and malignant brain tumors, they're approximately as common as leukemia in men, and then slightly more common than melanoma in women. So we think of them as rare, and they are rare, but they actually do affect quite a substantial proportion of people each year. And again, I'm sure I don't have to tell this to this group, but brain tumors are a very heterogeneous disease. There's a lot of different types. There are some that are more common than others, particularly meningiomas and other non-malignant tumors. But for the purposes of this talk, I'm going to be focusing on glioma. So within malignant brain tumors, the vast majority, about 80%, are the broad category of glioma. Within that, slightly or somewhat more than half, or about 60%, are glioblastoma. So that often predominates, and when we're looking at glioma epidemiology, we're often focusing on GBM. But it also includes a diverse group of other subtypes, including, other kind of classic astrocytomas, oligodendrogliomas, and then some other rarer histologies. So one of the kind of common observations that we have about cancer in general, and also in glioma, is that incidence of these tumors vary by age. So overall, we see increasing frequency of glioma, particularly glioblastoma, as people age, where we really see the median age is about 65. So the majority of people who have glioblastoma are in that elderly population. If you look at some of the lower grade tumors, such as the lower grade astrocytomas or oligodendrogliomas, you do see a slightly different distribution where they are can be more common. They have a bimodal distribution where they're common in younger ages and then also in older ages, such as lower grade astrocytoma, or oligodendroglioma, which usually affects people most in middle adulthood. And in addition to that age-specific pattern, we do see throughout the lifespan that these tumors occur more commonly in men than women, and that age difference usually increases with age. One of the other common observations that we have about these tumors is that while they are more common in men, men who are diagnosed with glioblastoma generally do poorer than women. And we see this in kind of national data, we see this in clinical series, and you continue to see it after you account for things like changes in, or differences in performance status, differences in treatment. But interestingly, we really don't see this in lower grade tumors. So these are from 2 different papers that we did looking at GBM, including specifically IDH wild type GBM, and then on the left, and then oligodendroglioma and lower grade astrocytoma on the right. And you can see on the right, there's really no difference. We don't see a female survival benefit overall in that population. But when we look specifically at the relationship between age and sex and survival in glioma, we do start to see that there are patterns here as well, where both the survival benefit by sex varies by both age and histology, where we see that biggest female benefit for survival in that kind of middle, early adulthood. And then as people age, we really don't see that survival benefit anymore. So there is some kind of more complex interplay, I think, with the impact of sex on survival in these tumors that we really don't understand. One of the other projects that we did, and this has also been observed by others, is that higher socioeconomic status is associated with increased risk for particularly glioblastoma. This is based on an analysis we did in US looking at county level socioeconomic status based on measures from the American Community Survey. And so we found a significantly increasing trend of glioma risk in counties that were higher SES. This has also been replicated in other locations such as Scandinavian countries, where they have much more rich linked data sets and more information about educational attainment and things like that. But we do still see it in the context of national health systems as well, suggesting that it's not necessarily just related to access to care. One of the other consistent observations that we see with glioma is that it's most common in individuals who have are in the US would be non-Hispanic white, or would be generally grouped as kind of a European ancestry population. So if we look internationally, we see, this is a heat map, this is from data from the World Health Organization, that the highest frequency of astrocytoma is in these dark purple countries. So it's North America, Western and Northern Europe, and then Australia, and with lower frequency of astrocytoma when you look at other locations in the world. And we see the same pattern if we look in the US by race ethnicity, which is kind of a crude, measure of looking at genetic background or those differences, but we're able to see repeatedly in all glioma types that we have the highest frequency of these tumors in individuals who are white, non-Hispanic, with about 30% lower incidence in individuals who are white and Hispanic, and about half the incidence in individuals who are Black or African American. And we see this in all glioma types, so it's fairly consistent. And in particularly with glioblastoma, we generally don't think that there's a significant access to care issue and whether or not people are diagnosed with that tumor, because it's so severe that we don't, again, much like SES, don't necessarily think this is an access to care issue. And then following from that, one of the analyses that we did was look at a large, well, this is actually from 2 papers. So we looked at a large population of adults with glioma that were self-identified as Hispanic or Latino from 2 large studies, one centered out of Houston and then one centered out of Tampa in Florida. And we looked at their genetic estimated proportions of American, African, European ancestry. And we found that individuals who were diagnosed with a glioma in general had higher proportions of estimated European ancestry as opposed to individuals that were not diagnosed with a glioma. And this was in adults with adult type diffuse glioma. And then working with a collaborator on a study looking at pediatric ependymoma using data from California, they found the same pattern in children, Hispanic children who are diagnosed with pediatric ependymoma, that on average, they had higher estimated European ancestry as compared to controls without glioma. And this group has also found the same thing in pilocytic astrocytoma. So it does seem like there is something there about European ancestry in particular that predisposes people to be diagnosed with glioma. And then related to this, we've also recently did a study looking at self-reported his origin for individuals who are Hispanic in the US. So we looked again at American Community Survey data and looked at whether or not individuals in the county more commonly reported that their ancestors were from the Caribbean. So that would be Dominican Republic, Cuba, Puerto Rico, as opposed to Mexico or Central America. And so we dichotomized counties based on which of those was the most common reported ancestral background. The reason for this being is that there's a significant amount of evidence that the patterns of genetic admixture are really different in those populations. So people with a family history that originates in Central America or Mexico have much higher proportions of Native American ancestry, as opposed to individuals who have family origin in the Caribbean, where they have, in general, much higher proportions of African and European ancestry. And so we compared frequency of glioma in these two dichotomized counties. And we found that in general, frequency of glioma was lower among Hispanics in the US in counties with self-report where the most common reported ancestral origin was Mexican or Central American, as opposed to Caribbean. It was about a 20% decreased risk of or incidence of glioma. And this was consistent when we looked at large counties or small counties, as well as when we adjusted for things like frequency of in-migration, out-migration, to kind of account for the fact that people might be leaving the country or things like that. So it did seem robust to all those associations. So that fits with our other work suggesting that there is something perhaps about increased European ancestry that predisposes you to a glioma. So now to kind of, so that's just kind of like laying the groundwork for who gets glioma in the US. But so the rest of what I want to talk about is thinking about what risk factors predispose someone to get a glioma. So unfortunately, we really don't know any. There's no single risk factor that accounts for a large number of brain tumors. There are a lot of single gene Mendelian disorders that predispose people to develop a brain tumor, but it's a very small proportion of cases that can be attributed to that. There's an increase in association with ionizing radiation. There's some interesting associations with allergies and other immune conditions. And then there are some genetic variation that's associated with risk. So just briefly, ionizing radiation exposure is really the only known risk factor for development of a brain tumor. And it really has to be therapeutic levels of radiation exposure to the head. So the kind of hallmark study that identified this was this Israeli tinea capitis cohort, where while there was a significant amount of in-migration to Israel in the 40s, there was an outbreak of ringworm of the scalp. And so you had a lot of children with ringworm on their head that was treated with radiation. And so then you've ended up having this cohort of children where they were irradiated and their non-irradiated siblings. And so they've been able to really establish that that significantly increased risk of brain tumor in that population of kids that were treated with that. And there have similarly been studies in childhood cancer survivor cohorts that have showed the same thing. But the effect of this really requires that high level of therapeutic dosage, and it is most significant effect with younger age. So if you are less than 5 at the time of radiation exposure, you can have about a 30-fold increase of risk of a brain tumor, whereas if you're 10 to 20, it might just be 5 times. So it really is dependent on what kind of delicate stages of development you're at with the brain. The other very commonly identified factor is that history of allergies, particularly respiratory allergies, is associated with decreased risk of glioma, and it's about a 30% decrease. So we found this in studies after people were diagnosed, where we're asking them about their history of allergy. There have also been studies from large Scandinavian biobanks where they've identified that higher levels of IgE pre-diagnosis are higher in controls as opposed to cases, so that lower levels of IgE seem to predispose you to develop a glioma to some extent. And then following up with that, one analysis that we've done recently was look at the kind of the genetic predisposition to atopy or allergies, and then also autoimmune disease. And so we found broadly that genetic factors that predispose you to develop one of these immune conditions, such as, eczema, asthma, Crohn's disease, celiac disease, seem to be associated with decreased risk of developing a glioma. So we have, evidence from reporting, and then also some suggestion that it might be related to some sort of genetic susceptibility where they have anti-correlation. Then one of the other studies that was done more recently found that prior use of aspirin therapy is associated with decreased risk of glioma. So that would be, long-term treatment with baby aspirin. And this is from a meta-analysis of several large cohort and case control studies, and it found that not only does it decrease risk, but that we see increased protection with longer use. To the point and where over 10 years of baby aspirin use is associated with over a 50% reduction in risk of developing a glioma. So in addition to this, there have been a lot of other things that people have looked at. So all of these are at this point largely unproven, although that doesn't mean that they aren't, associated with it. It's just they haven't really been able to be linked. So there are things like head trauma, diagnostic radiation, cell phones have been historically a big one, and then a lot of other parental factors, which are most relevant usually to kids' brain tumors, but have also not been associated with most development of brain tumors as an adult. So the next factor that I had mentioned I wanted to touch on briefly was what we know about genetic risk for glioma. So there have been, over the last 15 years, several studies that have looked at families where multiple individuals in the family have developed a glioma, and they've identified several genes where rare mutation increases the risk of developing these tumors. So one that's very common is P53 with Lee Fermini, but then in addition to that, there also have identified significant associations with POP1, which is in the Shelton complex, and then also HERK2. Sorry. So then the more common type of glioma would be what we, in contrast to these familial gliomas, would refer to as sporadic glioma. So it's glioma that occurs in someone with no family history. And to date, we have identified 25 different kind of common genetic variants. So they occur in the population at a frequency of over 5% that affect risk of developing a glioma. In combination, these account for about 30% of genetic risk for glioma. So there's still a lot that these don't account for. But it's likely that other sources of genetic risk may be less common or have lower effect size or otherwise might be difficult to detect using these methods. So at the The variants that have been affected by these studies have pretty significant subtype differences. So the variants that increase your risk of developing a GBM and increase your risk of developing a lower grade glioma are quite different. So the only variant that seems to affect both equally are variants in P53. But other than that, they're quite different. So in particular for GBM, you have common variants in TERT and RTEL1, and then also in EGFR that predispose you to develop a glioma. Whereas in lower grade glioma, you have the most common variant is one in CCDC26, which is a non-coding RNA near MYC that significantly increases risk of developing, particularly an oligodendroglioma. And then you also have other variants in IDH1, another variant in RTEL1, and then variants in CD, KN2B, and CD and KB2A. I'm so sorry. So one of the significant limitations of these studies, though, is that they were done on data that was collected prior to the 2016 WHO change, where IDH mutation and codeletion status became critical to really subtyping these. So that's why, when you look back at those, they're just called like GBM, and then we often refer to that other group of just mixed LGGs as non-GBM. So there have been some attempts to kind of go back and look at these variants within the subtypes that have emerged. And so these are using the kind of the subtypes that came out of the TCGA papers. So these aren't even necessarily these aren't the classifications really that people are using now, but there are significant variation in the effect of these on specific subtypes. So, the TERT polymorphisms really only increase risk of IDH wild type tumors. This CCDC26 polymorphism that I mentioned really only increases risk of IDH mutant codel tumors. But then you have, the TP53 locus, which increases risk of everything. So then one last comment is that if you had been looking at some of those gene names, you might have noticed that there were a lot of them associated with telomere function. So you have POT1, which is part of the Sheltering complex, RTEL1, TERT. All of these are have polymorphisms that predispose you to develop a glioma. And actually, if you look at measurement of telomeres, individuals with longer telomeres are more predisposed to develop a glioma as opposed to those with shorter telomeres. So there is some association there. So it's not necessarily just a coincidence that there are a lot of telomere-associated SNPs. It's kind of sum things up. There's a lot to do in brain tumor epidemiology. There's a lot of things I mentioned here where I say we don't know things or that there's more to do. There's a lot to be done with improving risk factor measurement. So I think, thinking about how we're collecting data, figuring out how we can abstract data better out of existing data sources, there's a lot of reclassification going on. So, I mentioned a lot of these are using fairly crude diagnostic categories that we don't use anymore, but people are going back to reclassify individuals from these large studies so we can actually look at them in more clinically relevant ways. And there's a lot of ongoing initiatives to look at the effect of sex. So, if you go to SNO or things like that recently, there's a lot about, sex differences. And then also, I think, an increase in looking at the impact of, ethnicity, variation across different populations. Because a lot of these have really just focused on individuals who are white or European, just due to a lot of recruitment patterns where we do research. And I think that that's leading us to miss a lot about why people get glioma. So just to sum up, thank you. Hopefully this wasn't too fast and that it was interesting too. So I'm happy to take it.
Thank you. Thank you for your presentation. If anybody has any questions, please post them on the Q&A and we'll be happy to address them. I have a couple of comments. In terms of you're finding these populations that may have higher risk of specific diseases, right? So would it make sense or to think of a model that you would say if you have this type of ancestry or within this age group that you could start looking for these diseases, right? In terms of early detection, which obviously is not cost effective as a whole. But if you could do surveillance, there's some people arguing for surveillance for low-grade gliomas in normal populations. So that's one of the comments. And then second, there's been some studies, which I believe from your group as well, looking at prognosis that may be better, specifically, let's say in Hispanics compared to non-Hispanic whites and African Americans. And we don't know why. So if you want to.
Yeah. To comment on the screening question, it would be great if we could figure out how to screen people for brain tumors. As you mentioned, it's not cost effective. A lot of the focus, at least in people that I've worked with, is in those people who have family members with glioma, to try to figure out who should really be closely screened if they have a family history, because right now we don't know. And then there is some really interesting, I didn't include this, but there is some really interesting work that's come out looking at ethnic background, race, ethnicity, and survival. So we do see that individuals who are non-Hispanic white have the worst survival when they're treated with standard of care generally. It's not really clear why that is. I think that there are I think that there is, a lot that is potentially to be gained from looking into that, which some people are doing. We have some active research looking at that, that could be beneficial in general and help us learn more about how, kind of underlying genetic background, things that affects like the immune system, might impact certain glioma survival.
I really found it interesting that you having these allergies or having high levels of IgE reduces the risk of glioma, particularly because the brain is such an these antibodies really don't cross the blood-brain barrier. I wonder what is really causing or reducing the risk of gliomas in these patients.
Yeah. I mean, I think unfortunately, we really don't know. That's another area that I'm really interested in. I mean, I've talked to some of my immunologist colleagues about that. Because there are some other interesting associations with neurological conditions in general where that seems to affect prognosis. But yeah, it's interesting because, we really don't have the same type of immune response in the CNS as we do in the rest of the body.
Yeah.
Someone is asking about some of the work, if we can share the work. Most of the publications, are widely available and from the national databases as well. But we'll certainly share if you guys can questions, collaborations. Fantastic. Let me check if there's anything else. Well, I appreciate, just to respect everyone's time. We're going to keep these talks for about 30 minutes, and we'll be joining again the second webinar looking at the classification of brain tumors in 2 weeks. We're going to have Dr. Ballester from MD Anderson as a speaker. And thank you, everyone, and truly appreciate your collaboration and willingness to help Dr. Ostrom for the CNS and AANS organizational affairs, which really made this happen. Thank you, everyone.
Yeah, thank you.